I'd like to thank the organizers for this opportunity for me to present a look at Fujifilm Diasim development of an advanced gene therapy platform. Fujifilm Diasim is a world-leading CDMO. We have 4 different sites. We have one in the UK, one in Denmark, one in North Carolina, and one here in College Station, Texas. At our 3 sites in Denmark, the UK, and North Carolina, we specialize in proteins and monoclonal antibodies. We go all the way up to a 20,000 liter scale. We've actually had 14 commercial projects being launched by those sites. We've run a total of 325 different projects. We've got over 25 years experience, and we are quite experienced in different types of technologies. Here in Texas, it's primarily virus-based therapies. This is a look at our College Station site. What you see here is that it was founded in 2011 by Texas A&M University as K-Line Biotherapeutics, and it was acquired by Fujifilm in 2014. We have actually 2 sites here. The first site is called the NCTM, or National Center for Therapeutic Manufacturing. The picture on the right shows you what that site looks like. It utilizes mobile clean rooms, which are individual units that have high HEPA filtration in and out so that the air is completely sterile. These can be configured in different types of groups, 2, 3, or 1 unit, with a walkway between them that is also well controlled. And therefore, each one of these is a manufacturing facility in itself. At the end of a process, we can vapor hydrogen peroxide treat these materials. And therefore, we like to say that the manufacturing site here starts off at a neutral situation, and then at the end of the manufacturing process, we bring it back to a neutral situation. We have a second site called the Biomanufacturing Facilities, which is made up of 2 buildings, and that's shown on the bottom. On the left is our monoclonal antibody production facility, where any antibody that follows our normal Saturn process is produced here in Texas, and we have several different 2,000 liter trains for producing those antibodies. The right half of that figure is our biomanufacturing viral section. And as you can see in that figure, there are slots which are also doors in which these mobile clean rooms have been inserted. We have 5 currently. We have room for 10, and we actually are going to be expanding both of these facilities as we launch our new gene therapy expansion initiative. So the NCTM right now is configured to be able to take you from preclinical to phase 2. We are going to do some work on that so that we can actually extend that all the way out to commercial, and we'll use this as our smaller scale facility. So this will go up to 500 liters of manufacturing. The flexible biomanufacturing facility right now goes basically from phase 1 all the way out to commercial. And as I said, we can do multiple trains at 2,000 liter in that facility. And we will be continuing to expand that to allow us some additional capabilities there as well. So what do we work on here in Texas? When we talk about advanced viral therapies, we really basically talk about 3 different types of things. One is in vivo gene therapy, predominantly using AAV vectors. The second type of project is oncolytic viruses, predominantly adenovirus, although we have had a project utilizing a phage to target tumor cells. And finally, next generation vaccines, where viruses are specifically attenuated by deletion of certain genes and complemented on cell lines that produce those genes so that you have an attenuated virus propagated only on a cell line that supplies the missing function, and therefore replication incompetent in the individual that's receiving it. All 3 types of projects are something that we've worked on here in Texas, and we continue to do so. In terms of AAV production, there are 3 general systems that are used, and we've worked with all 3 of those here. We've worked with virus-infected cell lines, so we've utilized the baculovirus system, infecting insect cells. We have 2 different viruses, one encoding the rep-cap functions of AAV, the second virus encoding the gene of interest, flanked by the necessary elements for expression and integration into the virus particle. Second is a very similar system utilizing HSV, again, 2 recombinants, And there we performed that with Vero cell line. And again, those two recombinants have rep cap or gene of interest. Co-infection in both of those systems results in the production of AAV particles. The third type of system is the 3 plasmid transfection. That is made up of adenovirus helper plasmid, a rep cap plasmid, and a gene of interest plasmid, and it utilizes HEK293 cells or any other cell line that expresses the adenovirus E1 region. Now, our experience here at Fuji with both of these platforms is both an adherent and suspension. So adherent, we've worked with the 3 plasmid platform on a Hyperstack 36 setup, where we have developed the process for working with 50 Hyperstack 36 units at a time in manufacturing, and around 20 to 25 in process development. And we've actually run with 5 different projects using that system. In suspension, we've worked with baculovirus in SF9 cells at either the 200 or 500 liter scale, and we've had 4 different projects with that situation. We've done the 3 plasmid transfection system for 200 and 500 liter scale, and we've worked with 5 different projects there. And we've worked with the HSV system at a 500 liter scale with 1 project. You may have heard recently we announced a partnership with Oxgene where we licensed both their HEK293 suspension cell line as well as their plasmid system, which will allow Fuji now to offer clients the use of the suspension cell line and plasmids from Oxgene when they don't have their own materials and want to make AAV constructs. We are going to offer AAV serotypes 2, 5, 6, and later on 8 and 9. We will also have the adenovirus helper plasmid, and we will have a gene of interest plasmid backbone that we can clone any client's gene of interest DNA into. The adenovirus helper and the rep cap plasmids will be available off the shelf as GMP material, and clients will be able to purchase the volume, number of vials of material that they need to support their program with us. The gene of interest backbone plasmid will be available also. We will take the client's DNA sequence, construct the gene, clone it into that plasmid, and then we will prepare GMP material to support that. As a first generation, we're looking to develop a process that includes 500 liter upstream production in a suspension bioreactor, single use, and then a downstream platform for purification to allow us to work with all the serotypes that I mentioned and trying to have as consistent a platform as possible, still able to give us good separation of full versus empties. We will offer an analytics panel to specialize on being able to quantify full versus empty and to be able to do full characterization of the AAV material as we generate it. And a lot of this effort is being led by our new innovation labs and team members who are working very hard to develop this platform process. I wanted to show you a little bit about how we characterize the Oxgene materials in order to establish our platform. This slide shows you what happened when we actually grew up the Oxgene HEK293 cell line and froze it down in both the recommended freeze media from Oxgene and in a commercial cryopreservative. As you can see on the left, when we thawed out cells that were in the Oxgene freeze medium, they came out at a very good viability, but over several days, they lost some of that viability. We had to do a refeed in order to get them to come back up to a high viability, but then they did behave properly and they were able to grow. And as you'll see, we were able to use these in order to demonstrate that we can make AAV. When we took cells that we had preserved in our commercial medium, they came out also at a very good viability, but they maintained that viability over the course of multiple passages. So once again, we were able to generate enough cells to do some of our testing of the cell line. And in both cases, really the doubling time was 25 to 29 hours. This is some data that we generated utilizing the AAV5 plasmid system from Oxgene. So we transfected both the Oxgene cells that came out from the commercial cryopreservative, as well as the cells that were frozen in the oxygen DMSO media combination. You can see that in the cryopreservative, the yield of genomes per mil was 2.24 e⁻11th. Total particles was 6.48 e⁻11th, giving us a total particle to genome ratio of 2.9. Cells that were frozen down in DMSO gave us 1.75 e⁻11th genomes per mil, total particles of 4.72 e⁻11th, and therefore a ratio of 2.7, very similar to the cells that came out of the commercial freeze. When we compared that to a commercial cell line that we also have in the lab, we found a similar result, 2.45 e⁻11th genomes per mil and 6.55 e⁻11th total particles. So again, we got a ratio of about 2.67. So the two cell lines did behave very similar with the oxygen plasmids. And what we concluded from this was that both cell lines gave comparable yield with the oxygen and commercial plasmids. I didn't show you the data for commercial, but it was actually substantially less. The oxygen plasmids gave over 1 e⁻11th full genome on both cell lines. The commercial plasmids that we utilized were only over 10 to the 8th genome copies per mil on both cell lines. We'll switch gears now and tell you about the rest of our platform that we are putting together. So the first generation platform will attempt to develop a 500-liter suspension cell platform. We will use a number of different sized single-use bioreactors to develop that process. We'll start off in shake flask and make our way from 1 liters to 10 liters to, in our innovation labs, and then transfer it to our PD lab to move it up to a 50-liter scale, and finally to manufacturing for 500-liter scale. We have a number of additional tools that we can use in order to determine the optimal situation for transfecting cells. We have DOS-GIP, which can allow us to look at up to 1/50th or 1/10th of the final volume that we want to work with. We have even some very small bioreactors that are only a few mils, which allow us to look at multiple conditions at the same time. And of course, as I said, we can use the scale up of the single-use bioreactors. Our first generation downstream process will be very similar to what we've already performed for a number of different projects. We'll have a lysis step, which is probably important for most of the AAV serotypes we're going to work with, along with treatment with benzonase to remove any whole cell DNA and unincorporated genomes. We will then have a necessary capture affinity resin type step. We will compare affinity resins that are broadly specific, as well as those that are tailored for a specific AAV serotype to see which will work best with each of the AAV materials that we want to make, and try to come up with the best platform approach. Then we will utilize ion exchange chromatography to do a polishing step. We want to achieve the best removal of empty capsids from the material and give us out a final full capsid situation of around 70 to 80% of the final particle number. And finally, we will do UDF, UFADF, and other post-capture chromatography steps to concentrate material and change buffer to a final buffer that clients may be interested in. And we will then do stereofiltration to have our final bulk drug substance. We also will offer capabilities for final product using our Vanarox filling machine. In terms of initial platform analytics, we will have transduction assays to look at the number of GOI producing particles in our platform. We'll also look at total AAV particles by ELISA or HPLC, genome copy number by qPCR or ddPCR. We will look at residual whole cell protein using commercial ELISA. We already have qualified a benzonase commercial ELISA, which we can set up then to just verify with any final client's particular formulation to make sure that there's no inhibition. Whole cell DNA, we will perform by qPCR. We have an assay that we've developed that works with any mammalian cell line. We've already shown it works with 293 cells, Vero cells, HeLa cells, and A549 cells. This is a 2-replicon assay. The first replicon is about 130 base pairs in size. That it represents the total amount of whole cell DNA remaining in the product. The second replicon is about 250 base pairs. This replicon represents the requirement by many regulatory agencies, including WHO, for showing that you have less than 200 base pairs of host cell DNA, because they worry that larger segments could actually represent oncogenes. Our QC department will look at bioburden, pH, and osmolality also. I will also mention that for the transduction assay, we are working with our sister company, Cellular Dynamics, to evaluate cell lines derived from their stem cell clones so that we can identify cell lines that are most active in determining the infectious particles coming from different AAV serotypes. We also have a number of plans for future improvements of the platform. In upstream, we want to look at the transfection conditions to optimize them. We've been told that the oxygen conditions right now are not necessarily optimal. We'll be looking to evaluate that. We're looking at improvements in the media to get higher cell growth and better transfection efficiencies. We're doing some of that work with our sister company, Irvine Scientifics. And that will also include looking at higher cell densities. In terms of downstream, I mentioned that we will utilize a two-step process. The first will be affinity purification, and we'll be looking to see if we can find the right resins there and improve the binding and elution profiles using those resins, and the ion exchange chromatography to get the best separation possible of full versus empty. In terms of analytics, we want to quantify full versus empty and partially full particles. We also want to understand, when we say we have full particles, what else is in there besides the genome of interest, and do we have any fragments of the genome, et cetera. Mentioned already the novel transduction efficiency cell lines and working with CDI. And we will be looking at a second generation whole cell protein assay with our colleagues in North Carolina, utilizing mass spec for that. So at this point, I will stop and leave some time here for any questions, and thank you.